The hepatitis B virus, or HBV, infects nearly 300 million people worldwide. Every year, around 1 million people die from hepatitis B complications. There is still no cure for the condition. How does HBV enter the host cells? Scientists from all over the world had spent the last half-century attempting to answer this key question. But it wasn't until 2012 that Wenhui Li, an investigator at the National Institute of Biological Sciences, and his team discovered the virus's essential gateway to the liver, the HBV receptor.
HBV is tiny but with very complicated code, so-called the amyloid proteins. The amyloid proteins of HBV is much more complicated than most of the other viruses. The toolbox is limited. You have to start from the scratch in many aspects. We tried different approaches and made every effort to overcome the obstacles, expected or unexpected, to find the receptor.
Back in the 1980s, a group of Chinese scientists found that HBV can infect a squirrel-like animal called tree shrew, making it the only species known to be susceptible to HBV other than humans and chimpanzees.
That was a truly remarkable discovery. Based on this finding and other pioneer works in the field, we were able to set up the infection systems in Petri dish in the lab.
Li and his team designed a viral ligand and developed a highly efficient purification process to capture the proteins that interact with the ligand. Eventually, they identified the candidate receptor, NTCP, by high-end biochemical analysis. Using genetic studies, Li shows that NTCP is indeed the functional receptor for HBV and its satellite virus HDV. The identification of NTCP as a cellular receptor for HBV and HDV immediately answers several critical and long-standing questions in the field. For example, why does the virus only affect the liver and not any other organs? Because NTCP is only expressed in the liver.
Why the monkeys cannot be infected by HBV? Because monkey NTCP is different from human NTCP by a few of the amino acids.
Li's discovery paves the way for new model systems for both basic research and drug development for hepatitis B. It has become clear that infection and reinfection occur in the liver of patients even under the most potent antiviral therapy. This cycle is crucial for HBV persistence.
We are working on a therapeutic antibody that breaks down this circle by blocking the virus binding to its receptor NTCP. We are delighted that it has shown some promising results in clinical trials.
For his discovery of the hepatitis B and D virus receptor, sodium taurocholate co-transporting polypeptide, NTCP, this discovery facilitates the development of more effective treatment for hepatitis B and D. The 2022 Future Science Prize in Life Science goes to Professor Wenhui Li.